Good evening, everyone. I would like to welcome you all to the sixth session of the Basic Modules in Infectious Disease organized by Federation of Clinical Pharmacists of India. I'm Dr. Alia Salim, Clinical Pharmacist, Infectious Disease Forum member of FCPI. I would like to welcome you all to today's session. So all these sessions till now, all the 5 sessions were bacterial. Today we are entering our kingdom of fungi. Before we begin, I would like to thank you all for making our virtual ID week a big success. I would like to thank all the speakers, all the participants. It was an amazing event. Um, so, before we move on to the session, I would like to go on through a quick introduction. The rules are all same. You can download your certificate. A feedback form will be made available in the comment box towards the end of the session. So if you submit it, you'll get your certificate within 7 days. And if you have any questions during the session, you can post it in the comment box below the video thumbnail. And those questions will be discussed in the Q&A session. And all the videos will be made available in the FCPI website. You can go through it later. And I think that's it. If you have any questions or queries, please feel free to reach us out at fcpaofficial@gmail.com, or you can just log in at that website. So without any further delay, I would like to welcome today's speaker, Dr. Saili Gore. She is a clinical pharmacist of in the Infectious Disease Department of PD Hinduja Hospital, Mumbai. And today's topic is the kingdom of fungi, basic microbiology and clinical implications. Saili, it's my honor to host you. Over to you.
Thank you, Alia, for your kind introduction. I hope I'm audible.
Yeah, you are.
So, a very good evening to one and all. I sincerely trust that you all are deriving a great constructive quality content from the derived modules so far. So we are back with another one. So my topic for today is the kingdom fungi, basic microbiology and clinical implications. So before I start, I just want to clarify that I do not have any relevant financial relationships with any commercial interest. So let's begin. We all know that in the realm of biology, living beings are classified and have been identified as different and diverse kingdoms of kingdoms. They are 5 kingdoms, namely Plantae, Animalia, Prostata, Monera, and the fungi. So they are the 5 principal domains with an addition of new kingdom of Archaebacteria, and they have millions of species and subspecies within them. So fungi has won a dominion over the fungi has won a dominion as how they have been predisposed from being the daily bread of bakery from being superior even than the bacterial world. So considering how the penicillin was derived from a fungus called Penicillium chrysogenum, which was a fungi, and I'm sure you all know the story of the great Alexander Fleming, a scientist who invented this penicillin. What he had done was he actually wanted to grow staph colonies, but he left the agar media to grow bacterial colonies. But when he came back, he saw a fungi which was grown on those colonies, inhibiting that staphylococcus. So this is how he derived the penicillin. This is how he invented the penicillin chrysogenum, and it was discovered that it can have the antifungal properties. So, so fungi have a profound impact on human health, biodiversities, and food security. They are the biggest recyclers. They maintain the soil fertility. They are also commensals and parasites that can be pathogenic to the human host. So, what exactly is a fungus? This word is derived from a Greek word which is mycus and which means mushroom. They are chemoheterotrophs. That means they live on the plants or the organic substances, whether they are living or dead, for their survival. They are saprobes, that is, they live on the dead materials. They are symbionts. Symbionts are the ones that um, benefit the organisms that they have been living with in close proximity with. And then they are also parasites that can be causing deadly infections to the patients. So currently there are about 1.5 to 5 million of fungal species, and almost 300 of them are clinically important and are known to cause infections. Their virulence and morphologic characteristics enables replications within the host. So they cause different kinds of diseases such as superficial infections, subcutaneous infections, systemic or opportunistic infections. So we'll discuss them in the coming slides. And as the fungal infections are increasing, the resistance of the pathogens to the antifungals is also on the rise. But the study of pathogenic fungi has actually received a scant attention, maybe probably due to benign but very serious nature of diseases and limited detection measures. So I'll just mention about epidemiology. You can see here that the fungal infections are just in the number of millions and billions. So globally, there are 13 million overall fungal infections, and almost 150 people die of those infections every year. So millions and billions of people are affected by skin fungal infection. They suffer from blindness. The patients who live with HIV or AIDS, there is a 25% mortality with fungal meningitis and pneumonia. Then 1.2 million people of fungal disease are actually misdiagnosed as TB, and there is an 80% of 5-year mortality in those. Then there are fungal allergies that contributes in patients with asthma with almost 50% of mortality. So 4.1% of Indian population actually suffers from fungal diseases, and overall there is no holistic estimate of the burden, but there is a geographical differences between the variations, which is a remarkable factor for this variability. So just imagine every day almost we breathe 20,000 times per day, and with amount of like 10 liters of air, and every 1 cubic centimeter of air has around 50,000 of fungal spores. So imagine how much we are exposed to that bug. So just to additionally let you know that the Candida albicans, Aspergillosis and Cryptococcus are the highest infections that occurs in the fungal diseases, and they have the highest mortality. So now let's begin with our main objectives of identifying the basic morphology, physiology of fungi, distinguish different types of fungi, differentiate between the fungal pathogen and the normal flora, understand their methods to identify or detect the fungal organisms, classify various infections, and host how our immunity plays a role in abating these infections, and shortly illustrate the pattern of resistance amongst the different pathogens. So first and foremost, I chose this WHO fungal priority pathogen list as to introduce these organisms to you. This is a catalog of 19 fungi that pose greatest health threat, and they are rapidly emerging as resistant pathogens from the clouds of bacterial resistance. So they're also responsible for the infections, which actually are have which have the treating challenges exist and are associated with high morbidity and mortality. So WHO just has systematically prioritized them into critical, high, and medium level of groups with an aim to strengthen the global response to tackle the fungal infections and the resistance. So, morphology is very important to give you a basic idea of how the the bacterial or the fungal cell structures are, and where the mechanism of drug action takes place. So how are these fungi distinct at cellular level? So there are some contrasting characteristics of these uh, because of their morphology and physiology, and then likewise they'll have different disease presentations. So here I've put some basic and distinct differences between a fungi and a bacteria. So the cell wall of fungi is eukaryotic, and that means there is a nucleus present in that. And the cell wall of bacteria is prokaryotic. There is no nucleus present in that. And there is a cell wall. Fungal cell wall is very rigid. They contain chitins and glucans. The bacterial cell wall is semi-rigid and they contain peptidoglycans. Ergosterols are present in the cell membrane of the fungus, while proteins and phospholipids is present in the cell membrane of bacteria. So the powerhouse of fungi is just like us. They have mitochondria, while mesosomes are majorly the powerhouse in the bacteria. Fungi are unicellular and multicellular, but bacteria are just a single cell. Fungal cells reproduce by both sexual and asexual methods, but bacterial cells reproduce only by asexual methods, and they both respire by anaerobic and aerobic modes of respiration. So, fungi do not move, they don't have pilus, so they can't move, but bacteria can move with the help of pilia. Fungi are a bit bigger in their size variations, while bacteria are a bit smaller in their size variations. Then the colonies of fungal organisms actually grow slowly, but bacterial colonies have rapid growth, and the colonies of fungal organisms appear rhizoid. They are irregular or filamentous. They also appear as coarse and rough, powder-like or thread-like, and they spread all over their agar plate. But the colonies of bacteria are fixed, circular, and they are very specific. They are circular or oval, and they appear very smooth, and they are very damp or glossy, and they don't spread. They're just stagnant. Wherever you put them, they grow there. And so the fungal organisms don't have any odor, but they're mostly earthy. They have an earthy odor, but bacteria have a strong and specific odor. So examples of fungus are yeasts and molds, and bacteria, coccus, bacillus, and spirochetes. This bacteria you must have already gone through in our previous modules. So this is how a basic fungal cell looks like. So they have this cell wall, cell membrane, cytoplasm, nucleus, mitochondria, uh, reticulum, and Golgi apparatus. So when you look at a fungal cell, what exactly are you looking at? Are you just looking at one cell? You're looking at multiple cells, or you're looking at buds? You're looking at yeast cells, or you're looking at the thread-like structures which are called hyphae or mycelium. What are they? So fungal cell appears mostly in two forms. One is this like a yeast cell, this where there is a bud formation. They either stay attached to this parent cell, or they separate and form a new budding cell. This is called budding yeast cell. And some other fungi appear in the form of hyphae. So they have this multiple hyphae that form mycelium, and the hyphae are actually structured into like one. There are some hyphae that have cross walls. They are called septate hyphae, and there are some hyphae that do not have cross walls over here. They are called aseptate or non-septate. So, the components of the cell are usually very important to understand the mechanism of action. So, let's go through the three components of the cell wall. Of the fungal cell. So first is the cell wall, which I already told that it is very rigid. Chitins are embedded in a matrix of proteins, lipids, and polysaccharides, and these polysaccharides are nothing but glucans. They are having varied proportion of mannose, chitosins, and galactins in different types of fungi. And the major component of the cell wall is beta 1,3 glucan that is actually responsible for constructing the cell. It also divides the septum and assembles the spores. So we all know that basically any cell wall provides rigidity, skeletal support, maintains its shape and prevents the cell from mechanical injury. It also helps in metabolism and ion exchange. So the cell wall is a primary target for echinocandin class of antifungal drug action. So cell membrane is not a cytoplasm or a periplasmic space. It is a cell membrane which is composed of lipids, proteins, carbohydrates, and ergosterols. So ever wondered what these ergosterols are? They're nothing but they are the combination of steroids and alcohols that are actually embedded in a bilayer of lipid. So what the cell membrane is responsible for that it regulates permeability of cell material, pathogenicity and virulence. It also regulates the hemostatic mechanisms and maintains the physiologic control. So this is how a cell wall is organized. You can see the manoproteins, glucans, and chitins, but this is a cell membrane where you can see the membranous proteins and ergosterol, which are embedded in this bilayer of the lipids, and they are distinct. They are very distinct. Their organization. They're organized in such a way that they have an hydrophilic, that is water loving heads pointing towards the outside, but hydrophobic tails, that is lipophilic tails pointing towards the inside. And this is the basic feature for the all the drugs, attracting all the drugs and water molecules. And this is how it this kind of arrangement helps the drugs transport transport through the cell and the cell membrane. Then microtubules, they are composed of protein tubulins, and they are usually only present in filamentous fungi, the fungi which has hyphae or the hyphal uh, appearance. Then they are basically essential for continuous and rapid growth. They are responsible for separation of chromosomes and intracellular transport of all these cellular organisms like vacuoles, nucleus, Golgi, and endoplasmic reticulum. And these are also a site. See, this is the microtubules here. They are responsible for the intracellular organelles transport, and they are the basic target for pyrimidine analogs like flucytosine or graciofulvin to act on. So let's move on to physiology. Cell physiology is very deep, but I'll discuss with you shortly. So, like any other living beings, fungal organisms also go through multiple pathways for their survival and adaptability. So I already said that they are heterotrophs, and they live on plants, chemicals, whether living or dead, for their survival. So they basically go through this main biosynthetic pathway of L-alpha adipic acid. And just like everybody else, like all the living organisms, they need oxygen, nitrogen, carbon, purine and pyrimidines and glucosamines for to facilitate the biosynthetic pathways. And the basic characteristic of a fungal cell is they have a strong underlying mechanism of genoplasticity and diversity that actually helps them to deal with the environmental stress. So how do they multiply? They go through both sexual and asexual modes of reproduction. In sexual mode, the spores are formed by fusion of two haploid cells to form diploid nuclei that undergoes meiotic division. So what is meiotic division? Is basically the chromosomes in the parent cell divide 2 times to form 4 haploid daughter cells, 4 single daughter cells. Then asexual mode is where there is a formation of spores, buds or fragments that take place, which is actually divided by that formation of buds, spores or fragments that take place, and they are divided by nuclei. The division of nuclei takes place by mitosis. So mitosis is nothing but the single parent cell divides into two daughter cells, including itself. So there are different types of fungi that exist, and they differ in all their structure, their ability to use the carbon, sugars, alcohol, and amino acids. And they are usually classified mainly on based on their reproduction. So they are fungi are classified into Phycomycetes. They are the lower fungi. They do not have septates, and they usually produce only the spores. Then there are other higher fungi such as Ascomycetes, Basidomycetes, and Deuteromycetes. They do have septate, but they produce conidia as well as spore. So this fungi imperfectly is only because the sexual phase of them is not yet identified. So they are kind of unknown. So uh, morphologic classification is required to be known more than the reproductive classification in clinical practice. So, because this will determine your line of treatment. So, fungi are usually there are three types, mainly three types of fungi. One is yeasts, molds, and dimorphic. But over here, I have separated the yeast and yeast-like over here just for your information. So what are yeasts? Yeasts are unicellular. They are just one cell. They adapt to body temperature. They appear as round and oval form in microscopy, and their colonies are very moist, creamy, and white color, opaque or colorless on media. So they reproduce only by asexual mode, and they produce buds, which actually uh, protrudes like this, and they separate from the parent cell. So these are the budding yeast cell. Example is only Cryptococcus or Saccharomyces or Rhodotorula. They are the actual yeasts which have budding yeast cells. But then there are some organisms. Okay, so I have this real-time images of all these organisms with me. Our microbiology, the doctors at microbiology department and the girls at mycology lab were very, very kind to share these pictures with me and resolve all my queries. So I've got this Cryptococcus picture over here on a guard plate. You can see that they have white and creamy colonies over here, but their size variation, there is a variation of size in their colonies. And this is a microscopic picture of Cryptococcus. You can see small yeast cells over here, white color yeast cells on this stain. Okay, now moving on to yeast like how are they different from yeast? They are same, exactly same as yeast, but the only thing is when they are they also reproduce by asexual modes, but they produce buds that protrudes, but they don't separate from those buds, they remain attached and then they form chains. So they form a pseudo hyphae. So this is how it looks. This is how a pseudo hyphae looks like. For example, Candida albicans, glabrata, all the Candida species, and Trichospora. So these are the pictures of Candida tropicalis on agar plate and microscopy, albicans, then crucii, and Trichospora. So you can see how their colonies are so similar. The agar colonies are so similar to each other. They certainly they will be similar because they are all yeasts. But and they do have this pseudo hyphae and also the yeast cells, which you can see over here on the microscopy picture. But if their colonies, all of them are same and all of them just look same, the Candida species, then how they are differentiated within themselves. So this is chrome agar plate, where the colonies on chrome agar plate yield some color. So Candida tropicalis yields a blue color, albicans yields aqua color, crucii has a purple color, and Saccharomyces is completely white. Now you must have seen some if you leave the bread or cheese open for a very long time, you must have seen some greenish thread-like structure appear on it. So they are nothing but they are just mold fungi. They are multicellular and they develop hyphae. So the hyphae are aseptate and septate, like I already uh, uh, explained about it. And multiple hyphae form one mycelium, and actually they look like a branching filaments which can be aerial or vegetative. So the colonies that appear on the agar plate are very fluffy, cheesy or woolly, and They reproduce by both sexual and asexual modes by forming spore as well as conidia. So, for example, Zygomycetes, Mucorales, Rhizopus, Aspergillus, Fusarium, and Pseudosporium. So, this is how a aseptate hyphae looks like, and this is a septate hyphae. So, septate hyphae, I already told that they do have cross walls between them. So, Mucor is one species, Mucor or Rhizopus species is one species that do not have septates, and their colonies usually differ from like the appearance of the colonies are from brown to gray. And over here you can see that they appear like a bulb like structure, and they have spores. They have spores on it and within them. And over you can see this Rhizopus homothelicus on agar plate and microscopy, and you can also see their mycelia on the calcofluor stain. Now, coming to the Aspergillus species, they are all this Aspergillus species, Aspergillus lentulus, flavus, and nidulans. So you can see that they have their colonies appear from blue to green color colonies, and they do not they have spores, but they have mainly conidia, which is attached to the end of the hyphae like this over here, if you can see, and they have a Y-shaped. Their hyphae are Y-shaped, while in mucor it's like a bulb structure. So mucor species is known to cause mucormycosis, and Aspergillus can cause pulmonary or disseminated Aspergillus infections. So this is another mold organism called as Fusarium. Fusarium colonies are usually pink or orange or red color colonies, and in microscopy they appear like teardrop shape or like leaves. And then this is Pseudosporium over here at the bottom. The colonies are completely white, and they do become golden or blackish as they age. So, coming to the third type, dimorphic fungi, they are thermal dependent, they are temperature dependent, which is a vital differentiation characteristics between the two. Dimorphic fungi grow as yeast and molds both. So they grow as yeast at 37 degrees Celsius and molds at 25 degrees Celsius, and they're usually endemic. So example are histoplasma, blastomyces, and coccidioides or Maturella or Sporotrix. So when do they change their structure from one form to the other is during tissue invasion. So there are some dimorphic fungi that exist as yeast in the nature, but they are actually mold in the tissues like Candida albicans. Then this is a picture of Sporotrix schenkii, where they're mold in nature, but they appear as yeast in the tissue. So you can see over here. And then there is histoplasma, which also appears as yeast in the nature, and you can see here the mold growth of histoplasma over here on the microscopy. Okay, so I'll just pose one question to you. Maybe we can allow some 2-3 few moments for having some answers in the chat box. So a 52-year-old female is suspected to have a fungal infection based on her clinical picture when she comes on admission. Her tissue biopsy was sent to confirm her diagnosis while the reports were awaited. And after a few days of the sample collection, we were very curious to know, like if you are very curious to know you can call up the mycology lab and ask for the provisional identification, and they can tell you. So provisional identification over here was informed that the agar media showed brown colored coarse colonies, and microscope evaluation showed non-septate filamentous hyphae. So which fungal pathogen is likely to have caused the presentation? Cryptococcus, Rhizopus, Aspergillus, or Histoplasma. So the correct answer is B, Rhizopus, because I already explained that they have non-septate hyphae, and their colonies grow in brown to black colored, brown to gray colored colonies. So this is a summary of basic and distinct features between all the fungal pathogens, where you can keep this as a ready reference to differentiate the organism appearance with you. So Candida, Cryptococcus, and Trichosporan all appear white and opaque colonies. Just that the Candida colonies are specific, but Cryptococcus colonies are variable in size. The Cryptococcus is distinctly identified by India ink stain, and Trichosporon is has urease positive, and they have a little rough colonies than Candida, and the test on germ tube is negative. So Rhizopus, they have no septate, they are dark grayish brown colored colonies, and they have bulb-like structure, while Aspergillus have septate hyphae, and they have blue-green colonies with conidial structure, conidial chain structure in a Y shape. And Fusarium are red, orange, or pink colonies, purple colonies. They are shape in microscope is like a leaf or a tear shape. Then Pseudosporium presents as white colonies, and they change their color to brown as they age. So uh, there is one more Pneumocystis girovecii, which is a yeast that is also there. Uh, it is a yeast like fungi, and it doesn't grow on agar, but it leaves black dots when the histopathology sample is viewed. Under a silver stain, it will have black dots like cysts. So coming back to the identification techniques, there are different methods that are used to detect the fungal organism, and specimen is collected on the specimen collection depends on the site of infection. So suppose there is a sorry. I'll just before we move on. Sorry, I'm so sorry. Before we move on to the identification techniques, I'll discuss about their habitat. So there are multiple fungi. Many fungi are actually present everywhere. They are present in soil, birds, animals, human flora, plants, and almost everywhere. And their hyphae, conidia, or yeast are the most infectious form of the fungal pathogen, and they are attributed to different kinds of infection at various sites. So this is distribution of commensal fungi and the resident fungi. So you can see that Candida is present in intestinal fluids, vaginal fluids, and oral, and it is also present on the skin. Then Aspergillus is a lung fungi which resides on the lungs. Then Saccharomyces is a food fungi, but it is also present in the intestinal fluid. And Malassezia and Candida are mainly the skin commensals. So what are commensals? So uh, commensals are nothing but those that inhabit the skin, but they are not pathogenic in nature, but can be termed as pathogenic when when the normal flora is disturbed or the immunity is compromised. So commensals organisms develops mechanism to colonize humans. We all are not free of bugs. There are just like staph present on the skin, even there is fungus also present on the skin, and they have the ability to survive over there as symbiont organisms, and they produce keratinase to digest hair, skin, and nails. So commensals are either residents or transient. Residents are the ones that are actually present on us since the birth, like they just live on us, like Candida albicans and Malassezia. They are not at all pathogenic but symbiont in nature. There are other fungi which are transient, like they are transient normal flora. They are derived from the environment, like Candida albicans, Tropicalis, Aspergillus. They are derived from the environment and they live on us. But the moment the immunity is compromised, they'll start turning into pathogen. And then there are pathogenic environmental fungi which are present in the environment, such as mucor or cryptococcus or coccidioids that are present in they all are present in the environment, like in birds or soil. So now coming back to the identification techniques, there are various methods that are used to detect the bug. So the specimen collection depends on the site of infection. So for example, if it is a superficial infection, the scraping of the nail or the skin or the hair is done. Or if it is a systemic or opportunistic infection, then the tissue is sent, blood sample or urine sample is sent for other investigations. So first is direct microscopy. Here, the specimen is directly collected and stained with different kinds of stains, such as potassium hydroxide, 10 to 40%. The 40% are usually used to identify the nail fungal organisms. Then gram stain is used to identify yeast. India ink is specially used to identify cryptococcus. It differentiates the cryptococcus organisms from all the yeasts. Then there are calcofluor, lactobacillus, cotton blue mount stains, and some more stains like acid shift stain, silver stain, trypan blue stain, and aniline blue stain that are used for histopathological identification of the biopsy tissues. This microscopy is also used along with the culture media for confirming the diagnosis. Microscopy will only identify, but culture media will help differentiate the organism. So different agar media are used, such as Sabrose dextrose agar, then cornmeal. Blood agar is used to identify cryptococcus or histoplasma. Then blood seed agar is used to identify cryptococcus, and chrome agar is used to differentiate the Candida species. Now, there are some difficult to grow. Some fungal organisms do not grow easily on agar media. So PCR is used. For example, it is used to detect pneumocystis jirovecii, which causes PCP, that is plasma cell pneumonia. Then there are some immunological assays such as antibodies identified by host antibodies that are identified by ELISA techniques. Then there are fungal antigens that are markers that are identified, such as BDG, galactomannan from the blood samples, or CRAG or histoplasma antigen from the urine samples. Then there are more automation techniques that are derived to identify the subspecies of yeast, such as Militov, white tech, or biofire. And then there are some more tests that includes radiograph images, such as CT scan of brain or lungs, where the tissue is been where the organ has been identified. Yeah. So basically, the brain or the lung imaging is done, which can also help us identify the mold infections like Aspergillus and mucor, where they develop cavity in the lungs. So let's study about the pathogenic fungi. Now you must be wondering that if we are so much exposed to all these fungal organisms, then why we don't acquire those infections on like all of us? But we do. Like there are some infections on skin, hair, or nail that can occur on normal individuals. But generally, in healthy people, the growth is discouraged by protective barriers. The protective barriers are the intact skin, naturally occurring long chain unsaturated fatty acids, then competition within the normal bacterial flora and epithelial turnover rate, where the epithelial cell actually contains cilia and that they remove the organism. Then mucous membranes that are covered with fluids containing antifungal substances. So, and then even if these protective barriers are breached, then the innate and adaptive immunity play a role. Uh, so this is how when the fungal organism comes in contact with the human cells, then the innate immunity is activated, followed by the adaptive immunity. So what happens is, innate immunity is a first line defense that is activated by the macrophages and the neutrophils, and the secretory immunoglobulin A complements. They go at the site of infection, site of the fungal entry, like that is called opsonization. They recognize the fungal organism, they go there and they ingest them and eliminate them. So that is called phagocytosis. So this is a basic first line defense mechanism that all of us go through, and this is enough to clear the fungal colonies. But if there is a breach in mucus barrier, or if the inoculum of the fungal organisms is very high, then or if there is increased exposure to the fungal organisms, then the second line defense, that is the adaptive immunity, gets activated, where T helper cell 1 and 2 produces response by activating the interleukin 2 interferon. They also activate tu- tumor necrosis factors, and they release cytokines that are responsible for the phagocytosis and killing. So T helper cell 1 produces protection response, and T helper cell 2 produces allergy response. Yeah, so basically macrophages and neutrophils are plays a role in killing the fungal pathogens, but it is the dendrite cells which helps us store the antifungal memory, and it also transports this fungal organism to this immunity army in our body. So that was the basic flow of the immune response. And for a disease to occur, now for a disease that occurs, there has to be an accidental or deliberate entry of fungi that happens through inhalation, ingestion, or implantation to inside the tissues. And once this happens, then there is a breach in protective barriers because of trauma, compromised immunity, or even disturbed normal flora. And when the immunity is not able to help eliminate the fungal pathogens, then they hematogeneously spreads at different sites even after getting phagocytosed. So that's how the disseminated fungal infections happen. And the disease mechanism is a bit complex pathogenesis, but they present as allergy, mycotoxicosis, that is a mushroom. There can be a mushroom poisoning once some people ingest mushroom and they get poisoning, or infections. And the severity of these infections depends upon the fungal inoculum, the magnitude of the tissue destruction, the ability to fungus to multiply, and host immune response. So coming to the second question, a 56-year-old male, bedridden since 3 weeks and has Foley's catheter in situ since then. He presents to ER with drowsy, generalized weakness, decreased appetite, but he has no fever. And on admission, his urine sample was sent. The catheterized urine sample was sent for routine and culture. So urinalysis showed few RBCs, 15 pus cells, 10 epithelial cell, and yeast. And the urine cultures came to be positive to Candida albicans. So what is the basic approach to manage this patient? So you can think, but I will still tell you the answer. The second answer is 2, because you can't start the treatment at instant. Why? Because Candida albicans can be a colonizer to this Foley because of the Foley's catheter in situ. So you don't need to start treatment over here at instant, but you can change the Foley's catheter and send the repeat urine culture to identify if there is a catheterized associated infection, because the patient has been presented with drowsy and to the ER, so he may have infection. If even if the Foley's catheter is changed and the repeat urine culture is sent, there is no need to start treatment, because this looks like colonizer, because even the epithelial cells are present in the sample. Okay, so let's so I'll quickly go through the clinical presentation of the fungal infections. So first, I'll discuss about the risk factors that precede to fungal infections. So, changing climate and threatened biodiversities, farming, agriculture, construction sites, working at construction sites, wearing occult clothing, trauma, wounds, and burns are some of the environmental, accidental, or occupational uh, uh, risk for causing fungal infections. Then, uncontrolled diabetes and malignancies are some predisposing comorbidities. Then others such as interstitial bowel disease, post-COVID sequelae, malnutrition, transplant, GI perforation and renal failure, and profound neutropenia are some of the main reasons that can be a reason for the fungal infection to occur, and even increasing in immunocompromised host. Then prolonged hospitalization, indwelling lines, dialysis, TPN, prolonged ventilation also can proceed to fungal infections. And even the overuse or misuse of broad spectrum antibiotics or misuse of steroids can lead to fungal diseases. So, what are the signs and symptoms? Often these fungal infections are very difficult to distinguish from bacterial infection initially on the initial stages, and they are usually misdiagnosed or they have a delayed diagnosis. And the signs and symptoms are like variable. The mild to moderate diseases may have skin rashes, skin discoloration, lesions, nail bed infection, or oral thrush that may be from mild to even severe oral thrush. Then severe infections presents with facial pain, headache, fever, hypotension. Myalgia is one of the symptom. Then there are also increased counts or leukocytes, and one distinct sign is increase in positive for galactomannan, that is gal or BDG, that is beta glucan, which is positive. These two come positive when there is a fungal infection that has been diagnosed. So, but BDG is usually false positive if many antibiotics have been used before, and they are absent in the mucormycosis. But galactomannan is present in aspergillus only. Galactomannan is positive, comes positive when there is an aspergillus infection, and BDG comes positive when there is a Candida or Cryptococcus infections. So, I'll quickly take you all through what kind of fungal diseases occur. There are many kinds of fungal infections, such as nail infections, vaginal candidiasis, ringworm, candida infections in mouth and esophagus. They are very, very common all over the world. Then blastomycosis, cryptococcosis, histoplasmosis occurs in those who travel or live in the endemic city where these infections are existing. Then aspergillosis, mucormycosis, candidiasis, invasive candidiasis are some of the infections which affect those with weaker immunity. Then there are some nosocomial fungal infections, and eye infection and mycetoma. Uh, so, they are mainly fungal diseases are mainly classified by the involvement of the different organs and the tissues, and they pose as very difficult to treat bugs because they share same cell biology like us. So I'll quickly take you all through the four different types of fungal infections. They are cutaneous infections. They are superficial and affects only the outer layer of the stratum, cornea, hair or skin, or even the nail cuticles. Then subcutaneous are they affect the tissues, keratinized tissues or bones. Systemic, they occur usually because of inhaling the cornelia or the spores, and that can disseminate as a systemic manifestation. Then opportunistic infection is very important to study. There are some human commensal or environmental fungi that turns pathogenic only when the immunity is compromised. So I'll quickly take you all through how these infections look like. So tinea versicolor, you can see there's a dark pigment, normal pigmentation is disrupted. Then there is black pedra where there are small black nodules that are stuck on the hair shaft. And then there are dermatophytosis. These are ringworms. Then there is mycetoma infection of the skin, and you can see there are lesions and pus discharges and swelling. Then sporotrichosis, which is also called as rose gardener's disease. Then chromoblastomycosis, where there is a dark and huge swelling and pigmentation. Then rhinosporidiosis, which has large friable polyps in eyes or nose. So they are subcutaneous infections, and these are the systemic infections like I already mentioned, coccidioids and histoplasma, which can cause as disseminated infection, or they can also typically cause pneumonia. Histoplasma causes pneumonia. Then there are candidiasis that can be from mild to moderate oral candidiasis. There's also invasive candidiasis. Then there is disseminated cryptococcus. This is the bad aspergillus lung you can see here, and the disseminated aspergillus infection. And then there is mucormycosis that causes retinal cellulitis or orbital cellulitis and blindness. So for the time constraint, I'll just skip this slide. This is a slide of Candida score, which is usually formed but not used routinely in practice. It is just formed to predict the risk of invasive Candida infections in the ICU. So there's this case where a 65-year-old male farmer with diabetes was taken to hospital with complaints of muscle pain, discoloration of skin on left foot a month back. He was admitted to the ICU with a picture of cellulitis, fever, and hypotension. Over there, he received peptides and clindamycin. He was also mechanically ventilated. He was deteriorated and he was referred to your hospital, where on admission you found that his HbA1c was very high, and blood culture was sent, and he was empirically started on Miro and TICO. So on day 3, his cultures were negative, but he has still spiked a new fever, and hence this patient is suspected to have a fungal infection. So what are the predisposing risk factors for this infection? So almost all of the above. His occupation, his uncontrolled diabetes, he was previously admitted some elsewhere with ventilation, and he also received multiple antibiotics. So these are all predisposing risk factors that causes fungal infections. So this is the last slide, and I hope you guys are still with me. And I just want to elaborate very shortly on the resistance mechanism of fungi. And despite there are several advances in preventive diagnostics and therapeutic measures, fungal infections are still prone to cause invasive infections, and they are associated with high morbidity and mortality. So it is very important to understand the resistant mechanism of these pathogens because it is essential to prompt the treatment in patients with fungal infections that are so deadly. So there are two types of resistance. One is microbiological resistance, and that confers non-susceptibility of fungus to antifungal in vitro. And there is clinical resistance that confers failure in eradicating fungus despite administering the susceptible antifungals. So fungi develops resistance in two mechanisms. One is intrinsic, where the fungi are even resistant even before the exposure, and acquired, where the fungi develops resistance through different mechanisms. So, for example, intrinsic are Candida cruzi is intrinsically resistant to fluconazole, and Candida albicans glabrata has now developed resistance to fluconazole. sorry, yeah. Okay, so these are the basic mechanisms for the fungal infections: modified metabolism, overexpression of efflux pump, genetic mutation, target site alteration, and biofilm formation. So Azole has actually developed four mechanisms, usually affecting the efflux pump, and there is the ERG11 gene is encoded, and it creates an overwhelming situation for all the antifungals, and also the ERG3 gene is also mutated. So all these mutations and the developed efflux pump causes decrease in drug concentration, and the target site is altered, and these enzymes are upregulated, and the antifungals are not able to confer their actions, and also they develop some other metabolism pathways. Then echinocandin resistance is less well studied, and it is still investigated, where there is mutations in FKS1 and 2 gene, and there is a cell wall stress that happens in this. And then Pauline resistance, there is defect in ERG3 genes and many other ERG genes. There is a cell membrane stress, and along with ergosterol, many other sterols are also accumulating in them, which causes Pauline to have complex with them, and hence they don't act. Paulines fail to act. Then there is flucytosine resistance, where the mutation in cytosine permease is responsible for causing the resistance. And flucytosine resistance develop very rapidly, so they are not used monotherapy. So this is one of the major setback, is that resistance of fungal pathogens in the environment, as they are already been exposed to broad spectrum antibiotics and antifungals in the environment. So change in pattern of fungicide use in agriculture and waste management, change at-risk human hosts, and change in climates may create a new landscape for fungal resistance. Natural disaster is also a novel route for fungal resistance. So, I didn't want to miss out putting up these slides where there are new names of fungal organisms that have been come up. So I won't read them because I'm still learning to pronounce them. So coming to an end, I hope you all have acquired some basic understanding of mycology, and there are some key takeaways for you all. Fungi are some distinct organisms, and there are mainly three types: yeasts, molds, and dimorphic. They are commonly environmental and commensals, but few can turn to pathogenic. And not only the antifungals, but the host immunity plays a major role in abating these fungal infections. And there are some risk factors that precedes to the fungal infections. And there are four types of infections that occurs, that is superficial, subcutaneous, systemic, and opportunistic. So resistance to fungi develops in various mechanisms, but like bacterial resistance, they do not produce any enzymes as a resistance mechanism. And although fungal diagnostics are a limited available resource in all facilities, rapid fungal diagnostics are advancing. And clinical pharmacists must be aware of all these microbiologic features in order to have a better understanding in managing the antifungal drugs. And this microbiologic information can help you all to lead an antifungal stewardship that is known to increase the quality of antifungal treatment. So then, in the upcoming modules, you will have the discussions will be on antifungal drugs, the spectrum, and the resistance mechanism in detail. So, and here are the references I put up for you all to download and read. You can read them. So I close with a warm thank you to all of you for your time and participation. I tried my best to simplify all the points and put up for you all, and I hope I suited my title of my presentation. Thank you.
Thank you so much, Siley. You made it very simple, very easy to understand. We have a couple of questions. Can I?
Yes. Yes.
Yeah. First question is, could you please explain identification techniques once again?
So identification techniques actually vary from different pathogens. Like different pathogens show have distinct characteristics, and different infections have require different types of identification. So if there is a superficial infection like nail or skin infection, then the organisms are directly scraped from, there is a skin or nail scraping done, the samples are collected and then they are identified under the microscope. And then some organisms do grow on the agar media, so they are also incorporated on the agar media. The agar media shows growth of many fungal pathogens under some temperature regulations. For microscopy identification, there are many kinds of stains used to identify under the microscope. So different stain can identify different organism. Like I mentioned, India ink is specifically used to identify Cryptococcus. Uh, then there are some other fungi that different stains like alanine blue stain or some acid shift stain that actually identifies PCP pneumocystis organism. Then there are some other identification techniques such as uh, uh, Malditov or Biofire or Vitek that differentiates the Candida species. It's used to identify the Candida species. And yeah, mainly these three are the main identification techniques used. Then there are some other features like chest radiograph or CT scan, brain imaging that can identify the fungal cavities in those organs. PCR and I missed one. PCR is also used to identify some fungal organisms like PCP pneumonia. And yeah, so BDG and galactomannan are two immunological markers that are used to identify fungal infections. Uh, so usually BDG is not present in mucormycosis. It is present only in Candida, and BDG can come false positive if there is high use of broad spectrum antibiotics. And it is the galactomannan that usually comes positive in Aspergillus infection. Sorry,
Thank you. One more question.
Yeah.
Which drug acts on pneumocytic gyroveli?
Sorry.
Which drug acts on pneumocytic gyroveli?
So pneumocystis gyrovecii actually has been treated by Septran. Yeah. It's an opportunistic infection where Septran is actually given. It is an antibiotic. It is actually given for PCP prophylaxis. So you will have a different session on antifungals. The spectrum and the action will be covered in those modules.
Okay, I think that's it. Thank you so much, Siley. Thank you for taking your time and preparing all the slides. It's really simplified.
Thank you.
And participants, once again, there will be a feedback form below the video thumbnail in the comment box. So you have to fill it to get your certificates. You can download it within 7 days. I think that.